Welcome everybody to another CRiO webinar. This is provided to you through CRiO.space, where we have an online educational system that is tailored towards the interested observer of the natural world. And to facilitate that kind of learning, we put together these webinars to share ideas, and hopefully these presentations are intriguing enough that you will feel compelled to visit CRiO.space and join us in our history of the Earth system. Today's topic is color vision. Color vision is something we often take for granted, but it has become so fundamentally important to the way we perceive the world around us that it permeates our culture and language. We describe people, for instance, as being green with envy, or seeing red when they're angry, or feeling blue if they're sad or sick. These are sort of funny examples, but they underscore, I think, from an evolutionary perspective, the benefits that color vision provides. Color vision is substantially useful from an evolutionary point of view. It helps organisms find food, by seeing, for instance, when fruit is ripe, and color vision can warn you of danger when spotting predators or even poisonous and otherwise dangerous organisms. Color vision is not something that all organisms have. And furthermore, color vision is not something that all mammals have. And this webinar explores something about why that is and evolutionary explanations for our color vision as humans. So color vision is useful in communicating. So, for instance, flushing of skin can be linked with certain emotions in humans, as well as, by the way, in other animals. Think about reactions that chameleons have, or octopi, versus certain stimuli. The range of colors, however, and wavelengths of light that humans can detect is from about 400 to 700 nanometers, as shown in this diagram. This is referred to oftentimes as the visible spectrum, but this is the visible spectrum for us, or human eyes. It's important to remember that many other animals are able to see wavelengths of light that are beyond what we call the visible spectrum. They can look well into the so-called ultraviolet, or into wavelengths, for instance, in the infrared range. In a way, then, as shown in the diagram on the right, with a human eye versus that of a bee, or in the case of a hummingbird, there are worlds that these other organisms see that are invisible to us, even though we share the same world. So the beginning of color vision is a very interesting story. But just, I want to underscore for you that color vision may be different from organism to organism, and even within a genus or a family of organisms, like for instance birds, there can be different kinds of color vision. So that certain birds, like shown in the left panel here, see wavelengths more in the near infrared, like cormorants, whereas there are other birds like hawks and jays that see light in the peak in the more short wavelength region of the spectrum. Jays indeed are able to see in ultravision in the ultraviolet area of the spectrum. And on the right shows a cladogram based on analysis of different gene sequences of different birds, showing a relatedness tree of the different ornithiform groups of birds: ducks, chickens, penguins, pelicans, parrots, wrens, etc. All of these have variable amounts of color vision that they inherited from their ancestors in the Paleozoic. There's a huge variety in structure of eyes across different animal groups, but there are common elements to all seeing organs. There are certain genes, so-called PAX genes, they're essential for eye formation. Photoreceptors of all eyes are made up of some combination of a vitamin A molecule named retinol and another photosensitive molecule called opsin, of which rhodopsin is a member. Rhodopsin is the red coloration that you see, for instance, in salt pans. When the water becomes very, very salty, a certain group of bacteria, archaebacteria, inhabit these salt pans, and they use a red photosynthetic pigment called rhodopsin. So both these genes that I mentioned, the PAX genes, and the opsins appear to predate the development of vision. So they're probably there for light detection from our bacterial ancestors. And like everything in evolution, these chemical adaptations for light detection were modified and incorporated for the new purpose of vision. And color vision is remarkable in its capacity behaviorally that permits animals to tell the difference between the spectral compositions of different things that reflect light, regardless of variations in intensity of that light. So bright versus dark, you can still tell the difference between, say, one color versus another, thanks to color vision. And this is interesting too. Color vision is widespread, although far from universal, and it varies from species to species and even among individuals within species. If you think of our human group, our single species, there is a spectrum, as it were, of color vision and of vision capabilities. To support color vision, visual systems had to emerge that possess two basic structures. First, a multiple sensor type structure, each providing a means for differential filtering of the spectral energy of reflected light or transmitted light, and comparators, something that can be used to contrast the signals originating from each of these different sensors. There may be many mechanisms that you can imagine that fulfills this basic structural need of having multiple sensors and a means to separate out these signals from these different sensors. The sensors of choice across all contemporary life forms are photopigments. But that is on this Earth and this life on this Earth. So there may be elsewhere in the cosmos different ways that spectral absorption is analyzed by the organism. So analysis of these photoreceptor opsins that I mentioned, gene analysis from many different species suggests that these genes belong specifically to all vertebrates in terms of groups of photopigments. And one of these is linked to the so-called rod pigments, whereas others are linked to cone pigments. And all four of the cone pigment opsin genes are thought to have emerged sometime early in vertebrate evolution, around, let's say, 540 million years ago. And that is shown here on the right, that color vision in this panel has a long evolutionary history. As I mentioned, it's more than 500 million years. And it is thought, but not proven, that this is something that emerged during the Cambrian explosion. On the left, you see different representations of branching times for different clades, like lamprey versus sharks, and then sharks versus sturgeons and paddlefish, gar, bony fish, coelacanths, then salamanders and newts with at about 320 million years ago with the amphibians, then frogs, toads, lizards, birds, and then mammals on the bottom. I bring this up because the history of color vision is intimately tied to the history of the Earth system. And we continue to live in the echoes of major upheavals in the Earth system, in particular at the Permian-Triassic boundary, a big extinction event, which has affected all life, and it helps us understand why we have the biosphere on the contemporary Earth that we do. The first image-forming eyes are thought to have developed sometime around the Cambrian explosion, but there is no fossil evidence for this. It's speculative. Color vision, however, is different from image-forming eyes. That seems to have developed somewhat later. In the absence of a good fossil record, it's impossible to determine exactly when this development happened. Apart from some parts of optical systems, such as that of the trilobites, which have these fantastic calcite lenses that cover their eye regions, other soft tissue components of eyes are one of the first things to decay after death, and therefore are almost never preserved in fossils. However, the earliest direct evidence of color vision was found in this organism, Acanthodes bridgei. It's about 300 million years old. It's a small fish, about 4.5 centimeters long, and is shown in the right panel here. Here's a fossil of this fish as reported by Tanaka et al. This is remarkable because this shows earliest evidence of color vision. And this was a fish belonging to a group of spiny sharks. It was widespread on Earth between 400 and 280 million years ago. This particular fossil reported here was from the Upper Carboniferous, about 300 million years old, found in Kansas in the United States. The fossil showed skin, articulated cartilage, and well-preserved eyes. Within the eyes, they found rod and cone structures, which affirmed the interpretation that this organism had color vision. In animals today, of course, things like birds, reptiles, fish, typically have 4 types of cone receptors and 1 type of rod receptors. Most mammals, and this is where things get really interesting, typically have 2 of these cone photoreceptors, not 4. Why? And within the mammals, the exceptions to this are the primates, including humans. We have 3. So animals, including this ancient fish that I mentioned, started off with black and white vision, right, and color vision. And somewhere along the way, mammals lost most of their color vision capabilities, or their wide color vision capability. And humans and other primates got a little bit of it back in a different way. So a closer look at different cones seen in animals gives us some insight onto how these different arrangements developed. I mean, birds have the ability to see red, green, blue, and ultraviolet light together. So they can see in higher resolution than humans. And human eyesight is quite good. Binocular color vision with good resolution and ability for attention to detail. Cats see somewhat similarly to humans with red-green color blindness, however. They see the world blurrier than we do during the day, but have very sharp vision at night, appropriate for the stem groups of all mammals, as we shall see, which are nocturnal. Humans have some of the highest resolution vision in the animal kingdom, but we have relatively weak vision at night because of this. Whereas snakes have low resolution color vision during the day, but they can also perceive light in the infrared. And snakes' vision is similar to what you may have seen in a thermal camera if you enter some natural history museums, that they can see body heat. And a cephalopod, such as octopi or squids, are completely colorblind, but they see in various shades of gray. They have very good perception of prey in low light and during the day. So although some have argued that the octopus eye and the human eye have similar structure, the function is different. And that is because of separate origin. Color vision in mammals, then. Mammals have generally just 1 group of rod receptors and 2 groups of cone receptors. The other 2 seem to have been deleted. So why don't we have a look at evolution of mammals to try to understand this? So the first true mammals evolved in the early Jurassic, as shown here in this figure, around 200 or so million years ago. From fossils that have been collected of that age, it's thought that these early mammals were small and spent most of their waking hours in nighttime. So these are nocturnal creatures. So they lived in the underbrush, in deep leaf litter, and so forth. Being active at night meant that there was more of an advantage to developing a visual system that could function in low light and that did not rely on color vision. So mammals appear to have lost 2 of their cone cell receptors during this early evolutionary stage when they branched off from stem groups and led to the mammals diverging from other vertebrates like reptiles and dinosaurs. This is in contrast to modern birds and reptiles. They are thought to have evolved along a different branch and retained complex color vision, which means that the dinosaurs almost certainly had color vision, and this is appropriate for their daylight hour lifestyle, and that vision must have been acute. So the distribution of, for instance, the cone color vision absorption peaks in birds and reptiles is also far more evenly distributed across the visible spectrum compared to that of primates, including humans. Old world monkeys, gibbons, gorillas, orangutans, chimpanzees, and humans, those organisms absent of a tail with color vision show kind of spotty coverage in color, and that's very different from birds and reptiles. So we are what are known as trichromates, so three color cone vision with three cone photoreceptors. They are not particularly overlapping in their peaks in the spectrum. The sensitivities of these receptors are noticeably different, however, from those found in reptiles and birds. Our photoreceptor peaks for the part of the visible spectrum that we see are quite close to one another, and the pigment that really is our main source of information for color vision is they're kind of grouped together, and then there's another pigment that has a peak sensitivity at a shorter wavelength in the sort of bluish region. You know, birds and reptiles have an additional photoreceptor to ours that is sensitive to ultraviolet light. They have ultravision, and it is possible to use a camera that is sensitive to ultraviolet light to get an idea of what birds and reptiles see, and patterns are revealed. But that would be an interesting topic for another presentation. So it's more than just cones. Birds and reptiles, 4 different cones, humans have 3. Other mammals have 2 or fewer, especially those who are nocturnal or where evolution has steered them towards developing vision in the low light of night rather than color. And why? Well, it's probably due to what is termed a nocturnal bottleneck at the end Permian mass extinction. 252 million years ago, something like 92% of species went extinct. And it's in this environment that mammals squeaked through, and maybe they made it through because of their lifestyle as nocturnal creatures, and also where they lived. So that major extinction event cut out any mammals that may have had the 4 color cones of vision and the ability to see in the ultraviolet. So in a previous webinar, we spoke about hair and feathers, and this is a related idea because nocturnal adaptations by mammals and the survivors of the Permian Triassic extinction event are carried over to all subsequent mammal groups. So what are some of these nocturnal adaptations that mammals had? Well, this bottleneck hypothesis is an old idea. In the 1940s, Gordon Lynn Walls described the hypothesis that mammals were mainly, placental mammals were mainly or even exclusively nocturnal. And that this is true for most of their evolutionary history, starting with origins about 220 or so million years ago, and that lifestyle persisted through the Mesozoic during the dinosaur era until the demise of the non-avian dinosaurs at the Cretaceous-Paleogene extinction event about 66 million years ago. Although some mammal groups, like ours, evolved to fill diurnal ecological niches, so being awake in the day and sleeping at night, the approximately 160 million years or so spent as nocturnal animals has left a legacy on anatomy and physiology of most mammals, and most mammals are still nocturnal. So adaptations by mammals for a nocturnal lifestyle have had a lasting effect on us. So we, like all other living mammals, have a history that goes back more than 200 million years. What are some of these nocturnal adaptations? That is the reason that most mammals do not have the broad spectrum of color vision. Well, one is whiskers. Whiskers are used in finding prey, navigating, socializing with other mammals, and this is something we discussed in the hairs and feathers webinar. So this is not the only adaptation, but it's the first one that really strikes out, is that hair is a sensory organ. It's not just for ornamentation, although it has become that. This is probably an adaptation to a nocturnal lifestyle, mainly related to the senses. So senses here, like an acute sense of hearing and the senses of smell are also well developed in the mammals. Most mammals have a large noses and a large olfactory bulb here behind the nose. Mammals have well developed senses of touch, particularly in whiskers, hair, but also on the rest of their body, like think about fingers. With the exception of some, the higher primates, the cornea of the eyes are exceptionally large. So that this gives a less acute visual image compared to birds and reptiles. Ours is much smaller and allows us to see things in finer detail and finer resolution. But all in all, mammals have very, very limited abilities in color vision. So another thing is that mammals are not very well adapted to exposure to the sun, as opposed to reptiles. This may be one of the reasons why we are endothermic, why we are warm-blooded. It enabled our early mammal ancestors to become less dependent on solar radiation and other environmental factors to warm ourselves. And this ability allows us to generate heat quickly in our bodies using this brown adipose tissue that exists in our bodies. Also, we have respiration rates that are very, very high thanks to our mitochondria, 5 to 7 times higher than those of reptiles of similar size. So it also requires us to eat a lot of food because we generate a lot of energy to power our function. There's also fur to assist not just in adding to our senses, but also to thermal regulation in night environments or in the cold. And because of our nocturnal lifestyle, we don't have a shielding mechanism against ultraviolet light, which is absent at night. You need to have daylight to have ultraviolet light, whereas infrared is something that is not sensitive to whether or not there's the sun out at that particular time. There's also we don't rely so much on visible light for things like navigation, nor do we need it for other photoreceptors, for instance, to indicate something about the cycle of the seasons. This makes mammals quite different from reptiles and birds. So there's also the fact that mammals have circadian rhythms and burrowing lifestyles. So circadian rhythm is a behavioral pattern in all basal groups. It's almost always nocturnal, at least in the placental mammals. We are an exception. I show this here. This is a circadian rhythm diagram for humans. So at 4:30 or so in the morning, we have our lowest body temperature. It sharply rises in blood pressure sometime after 6 a.m. at sunrise. Testosterone secretion, high alertness, best coordination. All of this is happening at peak daylight hours. But by the time nighttime comes around, melatonin secretion begins and deepest sleep is encountered. But for placental mammals, you have to shift this circadian rhythm clock somewhat dramatically to account for their nocturnal lifestyle. But burrowing, another interesting one, probably a reason why mammals survived so well in the Cretaceous Paleogene extinction, because as of today, there is no known burrowing lifestyle recorded for the dinosaurs. However, for mammals, a burrowing lifestyle allowed for sheltering from climate and predators during the day, as well as against catastrophic environmental change, like maybe the one posed by the giant impact that wiped out the non-avian dinosaurs. So color vision is inextricably linked to our evolutionary history as mammals, and it's something that's reflected in us every day. And I'm going to end by discussing something called the theory of color. So physiologists have long known that there's a kind of perception of color that by comparison between sets of colors. You may have heard about the color wheel of Goethe, and that's shown here on the left. That red versus green, blue versus yellow, dark versus light, colors of each pair being thought from a philosophical point of view antagonistic to each other, so that yellow stimulus is achieved by relative stimulation of red and green photoreceptors and so forth. So exposure to red causes a response in red receptors of the eye and inhibits response in the green. So you can see that in the opposite sides of Goethe's color wheel, shown here in the left diagram. And this helps explain the so-called formation of negative after images, when stimulus of one color is presented and the opponent color is seen when the stimulus is removed. So when you stare at a red object, and then you immediately look at a white wall, you will see a green representation of that red object that you were staring at. And so further investigations of the circuitry of the retina and pathways and throughout the visual cortex continue to reveal this intricate system behind our interpretation of color. And we can get an insight into our history as a species by looking at the connections that we as humans have to other organisms, all the way back to the fish emerging in the Cambrian explosion more than 520 million years ago. And that ends our webinar for today. I hope you enjoyed it. Visit CRiO.space for more information, and be sure to sign up for future webinars, and looking forward to meeting you again next time.